Okay, this is going to be 7A Part B, where the A part where we actually talked about DNA molecule as well as the history of determining whether or not it was the inheritance. This is going to be 7AB, where we're going to talk about the DNA structure and replication. I'll show you this video. Actually, I'll tag this video in the YouTube video later on, but it basically shows you how we go from a strand of DNA to a strand of chromatin to a chromatid, and then finally to a chromosome, which actually is held together by the centromere, and a telomere, which is actually the shoelace type thing that actually holds them all together at the end. Okay, so what does a DNA molecule mean? DNA stands for the deoxyribonucleic acid. Um, so we actually have this deoxyribose sugar along with everything else that makes it a nucleic acid. What's the function of DNA? DNA is the genetic code for building proteins. If you remember, proteins are everything that a body needs to make what the body needs. It also holds the directions for all the proteins that can be needed for a cell. And since life is made of cells, all life. What are the proteins used? That's a bad sentence. I should have changed it. How are proteins used? They're used for structure, like muscle fibers. They're also used for enzymes. They're used for transporting proteins, and they're also known for making hormones. Enzymes are probably very important in terms of the structure, in terms of making it inside the body. What's a DNA molecule made of? And I'm going to want you to draw this. It's got a phosphate. This is a monophosphate. Remember, we had triphosphates and diphosphates for ATP, ADP. This is a monophosphate. This is the 5-3 orientation. Remember, this is the way it normally is with a 5 sugar and a 3 sugar in this range. It is made of a sugar, deoxyribose, a phosphate, and it's also made of a nitrogen base. And this is what's going to determine whether it's an A, a T, a C, or a G, and we'll talk about that very soon. What are the 4 types? And then actually they are right there. Cytosine, urine we'll talk about when we get to RNA, or thymine, and these are called pyridamines. And then we have guanine and adenine, which are the purines, and notice these are the big ones. So A's with C's, G's with C's and A's with T's. When they actually bond together, when I show you that spinning molecule, this makes a larger nucleotide than what it does if we actually are putting the C, or the smaller nucleotide if we were putting the A. So we have what are called complementary base pairs. We actually have adenine along with thymine, and we have guanine and cytosine. And the greens are supposed to be showing you these green lines versus the orange, those orange lines. And then how do the DNA molecule base pair, how do they actually pair up? With hydrogen bonds. If you get an A and a T, they come together with 2 hydrogen bonds. And if you get a C and a G, there are 3 hydrogen bonds. But this is the section that you can't see between the molecules, and how one base pair right there actually does not really touch the other base pair between the A's and the C's, or the C's and the G's. A's and the T's, or the C's and the G's. How is a molecule shaped? It is a double helix, which means a twisted ladder, 2 twisted ladders. What makes up the sides of the ladder? That's the phosphate and the sugar, which makes up this silver backbone. And what makes up the rungs? Those are the nitrogenous base rungs. The adenine, the thymine, the cytosine, or the guanine. Base pairs, these guys are the backbones made up of the phosphate and the sugar, bonded with another phosphate and sugar, another phosphate and sugar. And then remember, we have a normal one and we have a reverse one. And the one here in the center, these are those nitrogen base rungs. And there are the hydrogen bonds, either 2 between the A's and the T's, or 3's against the C's and the G's. I also recommend that you draw this one. This is how we actually look at the molecule. And again, we have the phosphate and we have the sugar, which make up the backbone. We have the nucleotide and the hydrogen bonds, which make up the rungs. we have the nitrogenous base made up of a phosphate, deoxyribonucleic sugar, and the nitrogenous base. And we have the backbone made up of the phosphate and sugar. So these are the nitrogen base rungs, and this is the backbone that makes up the DNA, the twisted ladder, the double helix. And here's the 5-3 with the 5 carbon and the 3 carbon, 5 on top. Here's the 3-5 with the 3 on top of the 5, the upside down one. And remember, they have to do that. Much easier to do the 5-3, which is what's called the normal strand, and then the reverse strand, the 3-5. And the video, if you watch the video from 6A1, you actually saw that. The most amazing biological process. This is called the Central Dogma of Biology, how we actually take DNA and we from one gene and we copy it to DNA of another gene. It is called semi-conservative. If you notice, this one actually just gets directly copied, and then this one gets reverse copied. And I'll talk about that later. If you want to see a video, I'll put this video along with all these 3 videos down there. This explains the central dogma of biology, which is this process. Here's the one that's actually tagged with the first one, where we take chromatin and put it into chromosomes. And then here's DNA to mRNA, and here's mRNA to protein, which is the entire dogma, how we actually make a DNA into a critter. What's a DNA? What is DNA replication? This is when we take DNA and make a copy. Or we do that through mitosis, and we go from interphase all the way through. What does DNA need to replicate? To need to replicate, it actually needs to divide in mitosis. So if you think about the cell cycle again, we have mitosis, which ends making one cell into another, this one, and then we have an interface. We have the gap one, where we're actually copying different things. We have the synthesis, where we're making another set of DNA, which is what we do in DNA replication. And then we have the mitosis, where we actually take those and we line them up, and we separate them, and we make 2 new cells from mitosis. DNA replication, basically what we have to do is we have to unzip the ladder, then we have to take new bases and put them into both sides. And remember, we have one side which is the 3-5, and we have the other one which is the 5-3, and the 5-3 can do it just like this, and the 3-5 has to do it backward. Here is the DNA polymerase which unzips it. Sort of the helicase that unzips it. Here's the polymerase which actually puts the other base pair and actually makes another copy. DNA gets ready to uncoil. DNA unzips, and it's used as a template. New nucleotide base pairs along with the old strands, and this process makes a new strand of DNA. It takes the old strand and actually makes it into new strands. This is called semi-conservative. So if you look at the new strand of DNA, it actually has the old side and the new side, where this one has a new side and an old side. So it's called semi-conservative. It actually conserves part of and then creates the other part out of new. DNA replication, this is going to be a shockwave, which is not available on an Apple, but I'll put this link along with this video in the comments. And if you take a look at it, we've got C's and G's and A's and T's and T's and A's and C's and G's and A's and T's and G's and C's and G's and C's and T's and A's and C's and G's and T's and A's and A's and T's and T's and A's, and it keeps going on. And then we have all these things ACEs. These are all enzymes. They're going to go in and they're going to actually speed up the process or make it happen at a process that's a lot faster or slower than it normally would. And then we actually have this DNA binding protein, which is going to actually bind the DNA. So let's go take a look and see how this works. And we're going to start with the helicase, unzipping it, and then we'll actually use the rest of these to make the other one. It's going to tell me it's a warning, and I'm going to accept it. And I'm going to push play. And here we come with the helicase unzipping. There's the DNA polymerase. And then we actually have these other enzymes coming in and creating a brand new one on the 5-3 side. And then the 3-5 side, notice this is a much faster process. The 3-5 side's got to go a little slower. It's still read in a 5-3 direction, but it's backward. You notice this one's just about done, and this one's still going on. Then we come through and we fix everything. And then we need that helicase to come through and do the rest of it. But we actually make two new strands, the old strand and the old strand, along with the new strand and the new strand, semi-conservative. How does DNA repair itself? DNA, remember when we actually go through G2 actually looks at everything and makes sure that it's working okay. If it's not working okay, it tries to fix it. If it can't fix it, it's going to destroy the cell. But in G2, part of the process is looking at DNA and make sure that the DNA replication worked out correctly. So here's a problem with the DNA. It's called an AP site. And then we take this enzyme, glycolycase, and glycosylase, and this glycosylase actually moves along the strand that's bad, moves on to the AP site, and we actually remove the amino group of the molecule, that's deamination, and we take alkylation, and that's actually adding and removing the alkaloid molecule. But we're basically going to go up there and we're going to fix this thing. We actually identify it. It removes the damaged molecule. Then we have another enzyme come through, endonuclease, and it actually comes through, and these are really wildly animated type pictures, and it's not they don't really look like this. It's more of a regular protein with molecules, not these blobs. These almost look like they're ghosts. And that endonuclease comes in and actually removes the AP site. It also actually takes neighboring nucleotides and pulls those out. So we detect it, and then we go in and we remove it. And here comes polymerase and ligase, two more enzymes coming through. And they'll go through and they'll actually fix the gap. And if we're able to do this, it'll actually go through the mitotic cycle. And if we can't, the cell will get lysed. And that is the end of 6A. Thank you much for stopping by. Adios.